Well, hello, Dr. Malik. Today my presentation is going to be about mRNA technology and its future in this world. So first of all, what is mRNA technology exactly? So mRNA technology is basically a technology that depends on the usage of mRNA that is synthetically made by humans and inserting it into human cells so that these human cells use that mRNA to translate a certain protein. Well, how does it work? Well, first of all, we get the protein that we want to produce, we sequence it, we find out what amino acid sequence that this protein is built upon. We then kind of like reverse translate it into its mRNA code. Now, once we do that, we put it in a bubble like this. This is an mRNA strand. It's usually a single strand, unlike DNA, which is a double strand. We put it here inside a fatty bubble. We put it here, like this. You can see this fatty bubble in here. And then we mix it with a little bit of saline to kind of make the bubbles dissipate away from each other. And then we inject it inside the patient's cells. And then the mRNA gets inside there. And so what happens is that the mRNA gets in, diffuses through the membrane because it's covered in a fatty lipid. It passively diffuses in. It doesn't need no receptors nor gates to get in. It can just diffuse right away. It gets in there. Ribosomes come and aggregate on it. The fatty membrane kind of melts away inside the cytoplasm. And the ribosomes start translating that mRNA strands and start producing the protein that's encoded within. Now, with vaccines, we usually do that to translate spike proteins, as we can see here. These spike proteins of these certain viruses like COVID. We take that spike protein sequence and we make the cell synthesize it or synthesize the processed version of it. And then it gets shown on its MHC class 1 molecule. And so when immune cells come in, they recognize that sequence and they start making an immune response towards it, making memory B cells and T cells. And what that does is that it immunizes the patient from any future exposure to that virus. As we can see here in this picture, we can see that these are CD8 cells, usually killer T cells as we call them, and this is a cell that they are attacking. We can see here like these are toxic cytokines. The pros of mRNA vaccines are plenty. To mention a few, the first one is that they provide adjuvant activities to drive dendritic cells. So they're really good at making adjuvant activities. And what we mean by that is when we take the protein and we take the processed version of it and we sequence it and then we make it into an mRNA, your cell kind of does all the work. So we don't really have to add any extra adjuvants with the vaccine, unlike many traditional vaccines. So they're really good with when they show on dendritic cells, and they drive their maturation and thus elicit a robust T and B cell immune responses, as you can see here. mRNA is also intrinsically immunogenic with its ability to encode immune modulatory proteins. So usually, when they make the mRNA sequence, is that they also add the sequence for some immune modulatory proteins, which basically makes the immune system go a little haywire. We'll see that some cancers kind of manipulate the system a little bit, and so they make the immune system compromised by inhibiting the work of some of these proteins by competitive antagonism. Also, a third pro, and one of the main ones, is that it's actually cheap. And that's really important, especially since a lot of developing countries are the ones that get hit the hardest during any pandemic overall. And they're also relatively faster to make for different diseases, and that's going to come in handy for a lot of diseases, especially cancers, which have very differing antigens, as we're going to see in future slides here. Other prospects is that we can use them to make other vaccines. Like, for example, for a flu vaccine. There's usually no universal flu vaccine. Usually what scientists do is that they kind of predict a certain sequence for the flu vaccines and they make a vaccine for it. They predict some spike protein sequences. And that usually differs from one region to another. For, let's say, North America makes differing vaccines than, let's say, the Middle East, for example, because they have predominant serotypes there, predominant strains of the flu virus. Another prospect is that it can also provide long-lasting protection. Well, there's usually no vaccines that with long-lasting protection against malaria and tuberculosis. Usually people need to take booster shots every once in a while, and that can become a little costly and kind of, how can we put it into words, inconvenient for a lot of people. Now, especially with the malaria vaccine, malaria is predominant in developing countries, tropical developing countries. And as we know, vaccines are usually expensive. So that goes to one of the other pros of mRNA vaccines, which is basically the fact that they're cheap. Also, parasites like Chagas, elephantiasis, and hookworm and liver flukes usually have no vaccines. We usually just use antiparasitics. It's really not a big deal, but having an immunity against those will come in handy. Also, none of the viral threats that could become a pandemic, like NIF, Lassa, and Middle East respiratory syndrome. So, one of the problems is that we usually face is that developing countries are not really not what most scientists usually focus on making a vaccine for, because they're like developing countries and they don't have enough money to kind of buy the vaccines in bulk, and so they're really not worth scientists' time, and so usually they don't care about these diseases, although they are a huge problem for developing countries. But usually scientists don't really care for that because that's not going to bring them a lot of money. But with mRNA vaccines, it will become faster for them to make vaccines for these countries, and it will also be cheaper for the consumer to buy. Also, we have many other diseases that we have zero vaccines for, like Lyme's disease, the West Nile, the Zika, and hepatitis C. Also, none of the respiratory syncytial viruses, which kill infants, or have no vaccines for them. And that could be solved by mRNA vaccines. Now, finally, we're going to talk about cancer treatment. Now, mRNA vaccines can be really, really useful against cancers. And there's a lot of different ways that scientists tend to tackle this problem. And one of the ways is that they tend to search some of the spike proteins that are shown on cancers, and they try to find some common spike proteins so that they can make a vaccine that work with most people. And that proves to be a challenge usually, but there are some vaccines that are in trials currently against these, against certain types of cancers. Also, one way they usually tackle it is they make mRNA vaccines that usually inhibit some of the inhibitory proteins that cancers make to evade the immune system, which shows a lot of prospects so far. And that's basically it. Also, an extra piece of information is that, which I didn't really mention here because, well, I don't know why, but the HIV. Now, I've read some articles lately that there's a vaccine that's ongoing against HIV, and it's actually hitting really good numbers in trials right now, in clinical trials. And we're going to see how that goes. Yeah, that's it. Thank you for listening, and peace.